It's a delight to be here today, and as Paul said, I'm going to talk to you about something that's really near and dear to my heart, which is how do we put together information from the different senses? I know you've touched on this a little bit. I know Dr. Blake has given you a bit of introduction to this, but today we're going to take a bit of a journey into this topic that we call multisensory integration. Before I start, I always have to say that I'm at the point in my career now where I have been relegated to the role of chief cheerleader slash fundraiser slash spokesperson for an incredibly talented group of young people. And much of what I'm going to talk to you about today is really the fruits of their labors. I'm just the guy who kind of shapes the message around the corners. But this is some of the folks in my lab, and they are just a remarkable group of people. This is the magic of Vanderbilt. Always have to start with the obligatory cartoon. Okay, so hopefully for today's lecture you'll see that this is about the merging of the senses. And a little bit about me, a little biography about me. So I'm a neuroscientist. All of us that are teaching in this series are neuroscientists. And I'm a neuroscientist that has a very specific interest in perception. And for me, I had an epiphany when I was in graduate school. So I trained in a very large lab that was focused on vision. And in that lab, where you would stay up all night and do late night experiments in a dark lab and often hallucinate and see cockroaches on the wall. Sometimes they were there, sometimes they weren't there. It dawned on me as somebody who was fundamentally interested in perception that even if we understood everything there was to understand about vision, we were only going to get partway to solving the puzzle. Because in fact, we live in this world where it's not just vision. We're bombarded with information from the different senses. And so one of the major jobs that the brain has to do is it has to put that information together in a meaningful and a coherent way. There's a German school of psychology called the Gestalt School. I think it was referred to by Dr. Mayer last week. The Gestalt School is about this perceptual unity, about pulling pieces together to form a coherent perceptual whole. So again, the process of perception begins with the different senses. And I'm going to argue that our perceptual reality is really built on a multisensory foundation. That this world that we see is not a vision stream, not an audition stream, not a touch stream, not a taste stream. It's all of those things merged together into this seamless whole. It's a surprise. And actually, I'll tell you, as we get into the sort of the neuroscience piece of the lecture, neuroscience has been slow to really adopt this set of questions to really address this fundamental issue of how do we integrate information across the different senses. So I'm going to argue that we know a tremendous amount about vision. We know a lot about hearing. We know a lot about touch, taste, smell. But we know surprisingly little about how these pieces are put together. And that's sort of the theme for today's lecture. And again, I think all of us will completely intuit this slide, right? Well, obviously, I could have taken this in Times Square in New York, where we live in this world, an increasingly sensory world, where we're bombarded with information from different senses. So it just makes the challenge that much harder. Is how does the brain really not only decide what pieces belong together, but just as important, what are the pieces that need to be segregated from one another? Otherwise, the world would be extraordinarily confusing. Interestingly, although neuroscience has been slow to address this question, folks have talked about this for millennia. This is a slide from Aristotle. Actually, Aristotle didn't have PowerPoint, I don't think. So a drawing is ahead of Aristotle with a drawing of his. And he was already thinking about this idea of the confluence of information from the different senses. If you can see his slide down here, unfortunately, he got a little bit wrong. Where did all the senses go? They went to the heart during Aristotle's age. Okay? But he had the basic concept, this idea of integration, really sort of at the core of some of his writings. So people and philosophers have been talking about this for hundreds of years in the context of something called the binding problem. How do we bind the different pieces of our world together? My goals today, I have really three very, very simple goals. And if I convince you of these three, I've done my job. One is to take you on a bit of a survey to talk about why this multisensory process is so ubiquitous in our world, and why we believe it's so important for our perception, building our perceptual reality. Second part of the talk, I'm going to go into a little bit of the neuroscience behind this, a little bit about what we know that is happening in the brain that subserves this integration of sensory information. And then finally, if we have time, and I hope we have time, I want to talk to you about why we believe that, obviously this is a very fundamental, basic question. But my lab and a number of other labs are starting to really scratch away and show that multisensory function may actually lie at the basis for developmental disorders like autism. So hopefully we'll get to that towards the end. Okay, so first, I'm going to give you the, it's great to talk to an audience that's my age, right? When I go to the undergraduates and I talk about ventriloquism, I make reference to things like vaudeville. And I look at complete blank faces, right? They have no idea what I'm talking about. So you'll know what a good ventriloquist is, right? So I even, I've talked about the Borscht Belt, right? Some of you have heard of the Borscht Belt. So great act. For me, a wonderful illustration of this constant interplay that's going on between the different senses. So what a good ventriloquist does, he or she has the ability to do two things. One, minimize their articulatory apparatus so that the audible cues, their lips aren't moving very much. There are minimal visual cues associated with that. And then secondly, be able to move the dummy's mouth and head in concert with these minimally audible cues. Okay? And we all know that, we know the dummy's not talking. But nonetheless, we come away with this compelling illusion that, in fact, the sound source is coming from over here, not from here. So basically what happens is the brain, there's a physical translocation of where the perception of the sound source is. It's really here, but it ends up being over here. Think about it in the context of a movie theater, probably an even better example, right? Where are the speakers? They're all around you, okay? They're not on the screen. Nonetheless, we have absolutely no trouble ascribing individual voices to individual faces on the screen. Okay? Pretty amazing when you think about it. Dr. Blake, I think, showed you an example, talked to you a bit about the McGirk effect. All right, so you're a primed audience now, but I just wanted, for those who may not have been at this lecture, I want to take you through an example of this McGirk effect, which is, again, another illustration of this interplay between the senses. What I'd like you to do is, you're my experimental subjects for a few minutes. Okay, and I'm going to ask you to close your eyes, and this fellow is going to articulate a phrase, and your job is to tell me the phrase that you hear him saying. Okay, so let's try this. The audio, hopefully, is loud enough. So close your eyes.
Ba, 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 ba.
Okay, what did people hear? Okay, look at the screen. All right, you know the answer, right?
Ba, 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 ba. Ba, ba.
Same or different? Different. What did you perceive? Yeah, so the typical percept is da, D-A, or tha, T-H-A, T-H-A. Audio track's exactly the same, and you were all right. It was ba, ba. What we've done is dubbed over this fellow mouthing the phrase ga, ga. So in this circumstance, what's really interesting is the brain builds a new reality. Okay, it equally weights the auditory track and the visual track, and it doesn't choose one or the other. What it does is it merges them. And it decides on a new percept. In this case, the typical one is da or tha. These illusions are really, they're fun, they're great experimental tools. I actually use this illusion all the time in the lab to talk about these binding processes. But we can even use very, very simple illusions to sort of illustrate this interplay that's happening between the senses. This one doesn't work very well, so bear with me, but I'm going to ask you to focus first on the right side of the screen. Okay, and I want you to report to me. This is a visual task, all right? When we bring people into the lab, we tell them, your job is a visual job. You have to count how many flashes. So take a look at the screen. It probably won't work the first time because the timing is funky on this projector. But let's see if we can get it to work. All right, that didn't work, but how about this time? About now. All right, let's go over to the left side. Okay, look at the left side. Won't work the first time. Pretty vertical, right? 1 that time. Ambiguity in the other case, right? So I heard some of you say 1, some of you say 2, some of you say 3. And actually, when we bring people in the lab, the typical report on the right side is 2. It's always a single flash. All right, what we've done now is we've just surrounded that flash on the right side with 2 beeps. Okay, so those auditory cues are temporal anchors, and they fool the visual system. They basically tell the visual system, well, since there's 2 beeps, there must be 2 flashes there. Okay? Again, another really powerful tool that we use in the lab all the time. I'm going to talk today a lot about vision and audition because these are really the senses that we know the most about. But what I want to do is in the next couple slides is sort of illustrate that these multisensory interactions that I'm going to describe to you, they're not unique to vision and audition. Actually, you can see them across every single sensory system. They all talk to one another and influence one another in interesting ways. This is an example of what we call the visual vestibular illusion. I bring you into my lab, and I put you in a completely dark room. And what I do in the dark room is I project, in this case, a horizontal illuminated line, and your job is to tell me the orientation of the line. And when you're sitting still, not surprisingly, you do very well. You tell me that line is completely horizontal. However, now what we do, much to the surprise and the chagrin of those in the room because they don't know that they're even moving, is we start to rotate them very slowly on a rotational turntable. And as we rotate them, those accelerative forces associated with the rotation actually result in the perception of the line shifting to the oblique. The greater the force, the greater the acceleration, the more oblique. The line is always completely horizontal. But they will tell you at a certain point the line shifted to 45-degree orientation. Okay? Illustrating, again, powerful connections between the vestibular system in this case that's mediating that rotational processing with your visual system. One of the fun illusions, actually, I just learned about this recently. I was on a trip to Finland. In Helsinki, there's a lab, really, really good lab there that does a lot of functional imaging. I'll talk about functional imaging in a bit. But they came up with a super cool illusion. When they described it to me, I didn't believe it, and then they had me do it. And basically what they do is it's very, very simple. They have you rub your fingers together, okay? And if you just rub your fingers together, you know, you have a certain texture associated with that, right? A certain amount of smoothness or coarseness. Okay, well, what they do then is they put on headphones. You wear headphones. And now what they do is they put sounds in there of different frequencies, okay, high frequencies and low frequencies. And they ask you to continue to rub your fingers and report on the perception. And what happens is, as that sound frequency gets lower, the perception of touch gets coarser. You feel like you're rubbing sandpaper, okay? And as you start to go up into the higher and higher frequencies, it becomes like rubbing against glass, all right? So again, audition and touch. Really talking to one another in interesting ways. A fun one, if you want to scare your experimental subjects, is something called the rubber hand illusion. Okay, there's been a lot of work here on the rubber hand illusion. In this illusion, what we do is bring a subject into a lab, okay, and we have them actually just put their arms out on a counter in front of them. And now what we do is we basically put a baffle so they can't see their right hand. And now we put a rubber hand. Okay? Rubber hand, it looks like a rubber hand. Okay? It doesn't look like a human hand. It's a rubber hand. And now we start to gently stroke the hand of the person along with the rubber hand in synchrony with one another. Okay? And we do this for a minute or two. Okay? And the first thing you see is the people start to kind of get a little bit freaked out. They say, it's starting to feel like my hand. All right? But then you do the evil thing, right? You take a syringe tip and you jam it into the hand. And what happens is, first off, they pull their regular hand up, but then that little trace in the back is actually looking at a galvanic skin response. Okay, a physiological measure of the degree of anxiety that they have. They believe that this is their hand. Okay, there's an example of this. There's a guy who goes on the California sort of beach circuit. He does the rubber hand illusion, except he takes a hammer and he hits it on the hand. But the people there, you know, they're completely, I mean, within a minute or two, you really do take on ownership of that rubber hand, okay, just based on the synchrony of the movements that you provide. This is sort of my favorite example, right? It's probably everybody's favorite example. Flavor, right? I'm going to use the word flavor because flavor is a more accurate word to use when we talk about taste. Because if, you know, most of you probably know, flavor is a construct that is built on the basis of two senses. All right? So we know that we have this gustatory sense, the taste sense. Right? But the gustatory sense is really pretty rudimentary. It has 5 basic, what we call submodalities. Okay? Salt, sweet, bitter, sour, and one called umami, which is the taste of protein. Okay? That's kind of a weird one. Most of our flavor is built on the integration of that very simple taste or gustatory information with olfaction. okay, our sense of smell. We know this, right? You have a cold, what happens? Your sense of flavor and taste really gets dampened, okay? And that's because olfaction is a major player in this. Well, I will argue that our sense of flavor is built not just on the basis of olfaction and gustation, but actually the other senses play a major role in this as well. Okay? Asian cultures really take advantage of this. Texture is a much bigger component of the food and the flavor construct in a lot of Asian cultures. I have a good friend of mine who's he's a really wonderful psychophysicist, or psychologist, I guess you would call him. I wouldn't call him a psychophysicist. He would yell at me. His name is Charles Spencer. I'm going to tell you a story for about 2 minutes, all right? Because if you remember nothing else, you'll remember this story. And Charles he's at Oxford in England. Wonderful psychologist, has published over 1,000 papers. Very, very interested in flavor perception. And Charles decided, he got very, very interested in individual reports of flavor perception. Okay, so why do you taste certain things in certain foods and other folks may not taste those or may taste different things? So what Charles did is he decided that he was going to host a conference. He was going to host a conference in a chateau in Bordeaux. Wonderful place for a conference, right? Good choice there. And what he did is he brought 40 professional noses. Okay, these are folks who had collectively over 1,000 years of experience in the French wine industry. Okay, and the purpose of the meeting was to look at and evaluate the degree of concordance and discordance they had in evaluating the sensory characteristics of their wine. Okay, so Charles starts the meeting. He's a very, I can't do the stentorian, Oxfordian kind of, he's just really, wonderful in his emceeing of this event. Starts the event, talks about the purpose of the event, and everyone's sort of positioned at tables, and so graduate students come out and they bring out a flight of white wine. Let's just call it Sauvignon Blanc. And what Charles does is he says, okay, let's start to discuss what the different sensory characteristics are. And what happens is, over here, okay, there's a bit of an argument. Okay? There's a group, there's a table. Everybody agrees that there's citrus in this wine. Okay? But there are those who say there's an argument between tangerine versus lemon versus orange, right? And they're going, and Charles is feverishly scribbling away, right? Oh, blah, 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 blah. And then over here, there's a different argument, right? Everybody's talking about the mineral that they taste in it, but some of it is flint, and some of it is, they have all kinds of descriptors, things I don't taste in wine, right? But they're just arguing away, and it's a wonderful, wonderful thing. Then he does the second flight of wine, Chenin Blanc. Same kind of arguments break out, okay? Subtly different flavor profiles, so not surprisingly, subtly different arguments, okay? And then we build to the crescendo, right? Then Charles does what was really the purpose of the meeting, okay? Takes that flight of Chenin Blanc into the back room where his graduate students have red food dye, and they dip it into the wine, and they bring the wine out, all right? So chemically, the wine is no different, right? The red food color, red food dye coloring is not, in any way influencing the flavor. What happens? Okay, descriptions of the wine now. What do we hear over in this table? Currant, blackberry, licorice, okay? So, absolutely, and similar arguments over here. Total categorical shift in the descriptors. Okay, now these folks are talking about things that you usually ascribe to red wine. Again, chemically the wine is white. There's been an argument, well, actually Charles tells this story, he's very funny, he says, they immediately revoked his visa to ever travel to France again. And there's been a lot of argument in the literature that maybe these folks were even more cognitively biased to vision. But again, it's a very simple point that it makes, right, is that our sense of flavor is dramatically shaped, not just by olfaction and taste, but also by vision. Charles followed this up, and this was a really fun paper. It came out in Current Biology last year, which just, he does some really cool things. Some of you may have seen it. He showed, believe it or not, that your rating of how good a certain yogurt tastes is dependent upon the color of the spoon that you eat it with. All right? And that sounds totally random, except the mapping is beautiful. If you're eating vanilla yogurt with a white spoon, it tastes better than if you're eating it with a red spoon. If you're eating strawberry yogurt with a red spoon, it tastes better than if you're eating it with a white spoon, okay? pretty wild. Right? This is cool. These are the things that, you know, they get me out of bed every morning. It's like, I get a chance to study this. Randolph talked to you a bit, I think, about synesthesia. I just want to play you. I don't think he played you the clip. The context for this is, as somebody who's really interested in sort of the neurobiology of how does the brain integrate information from different senses, there's a group of people out there, arguments, still debate going on about how many folks actually are synesthetes, all right? David Eagleman, who's down at Baylor, suggests that it's probably about 1% to 2% of the population that has some degree of synesthetic experience. What these folks do is they have an abnormal association either within senses or across senses. I'm going to tell you about a couple examples, but first I want you to listen to, we had a conference here a year and a half ago, and I had a woman, Carol, who's a pretty prominent writer in New York, who's also a synesthete, talking about her experiences. And I'll just play you a clip of this. It gives you a good sense of what
I came back from college on a semester break, was sitting with my family around the dinner table, and I don't know why I said it, but I said the number 5 is yellow. And there was a pause, and my father said, no, it's yellow ochre. And my mother and my brother looked at each other like, this is a new game, would you share the rules with us? And I was dumbfounded, so I thought, well, at that point in my life I was having trouble trying to decide whether the number 2 was green and the number 6 blue, or just the other way around. And I said to my father, is the number 2 green? He said, yes, definitely, it's green. And then he took a look at my mother and my brother and became very quiet.
So again, the clip goes on, but the point of it is, severalfold. One is obviously to talk about that this is the most common form where there's a mapping between the color of the written word or numbers, or the numbers themselves and the color. So, and these are lifelong consistent things. So if Carol came back 20 or 30 years later, those mappings that she's sort of trying to still figure out are completely stable. This is an example actually taken from a book by Diane Ackerman called A Natural History of the Senses that ends with a chapter on synesthesia that talks about some of the more bizarre examples of synesthesia. This is a woman, believe it or not, every time she tasted baked beans, she felt a specific geometric object in her left hand. Okay? So, and the book goes on, and there's a lot of characteristic, a lot of really anomalous examples out there of these, again, strange associations across the senses. The contemporary thinking about synesthesia, we know very little about it, quite honestly. The contemporary thinking about it is that what happens during development, and you're probably familiar with this, is during development there's a lot of exuberance in the projections in the brain. Things go to the wrong places because they're trying to find their way. And then over time, with experience usually, what happens is those things retract back. And they end up innervating the target structures that you see in adults. One of the arguments behind synesthesia, and there's some evidence for it from imaging studies, is that what happens is that that normal maturational pruning process stops for some reason in individuals that are synesthetes. The other reason that I highlighted this example from Carol is you can see that there's a genetic element to this. It runs in families. Okay, she's talking about this conversation that she's having with her father. There seems to be, about a 10 to 20% transmission rate within families. So an interesting puzzle, again, just sort of, puts in context some of these interesting multisensory interactions. So that's the fun part. Okay, now we're going to talk a little science, but not too much science. I'm going to argue, so I come at my study of the nervous system from the perspective that these examples, these illustrations that I showed you, and I can give several lectures on these perceptual illusions, they're fun. But this isn't the reason that multisensory systems have evolved and developed. Okay? They've evolved and developed for very fundamental reasons. In this slide, from a science paper, this triptych really, to me, drives home what we believe the biological imperative of multisensory integration is. And we just take you through the panels. So this is to argue that in these 3 panels, what we have in this circumstance, so this is the classic dialectic, right? It's a cat and a bird. Cat wants to eat the bird for lunch. The bird doesn't want to be eaten. In the first panel, what you'll see is that the bird is chirping, but there's foliage. There's leaves in the way. And we would argue in this circumstance that this is auditory information in the absence of visual information. Okay? And cat does not detect the presence of the bird. Second panel, we flip the scenario. Okay, foliage is gone, but the bird is quiet. Now we have visual information in the absence of auditory information. Again, cat continues along its merry way. Really where multisensory systems, where the bang comes in, if you have both active at the same time, notice what happens now. Vision and audition, the cat turns, orients to the bird. If it's close enough, the cat gets lunch. Okay? So this is the real reason you get something for nothing in this circumstance. Neither one of the senses alone are sufficient to elicit behavior. But as soon as you pair them, suddenly now you get a behavior when you didn't have one to either vision or audition alone. We can see this in ourselves. Okay, this is data from my lab. We just run a number of subjects in something called a simple reaction time task. In a simple reaction time task, your job is very simple. You're sitting in a room, a dark room, and I'm going to present you periodically with stimuli, lights and sounds. Your job is press a button as fast as you can when you detect one of those lights or one of those sounds. This is the pattern of reaction times to that. Okay? It takes you about 160 milliseconds to respond to the auditory stimulus alone. It takes you almost 200 milliseconds to respond to the light alone. Anybody know, want to venture a guess of why you're slower to light than sound? Yeah, I mean, it's exactly right. It takes about 30 or 40 milliseconds for information to actually get out of your eye. Okay? There's so much complicated processing architecture within your eye itself that it takes a long time. So that's what makes up for that delay there. The important thing here is the yellow bar. The visual and auditory combination, you're faster. Okay? And this dashed line here, I'm going to come back to this a couple times. This dashed line is really important for me as a neuroscientist. Okay? Because what the dashed line is, it's the prediction. If the visual channel and the auditory channel came into the brain and stayed completely segregated from one another, you would be faster. Okay? And that's what that dashed line predicts, that you would be faster. But you're always faster than that prediction. Okay? And what that tells me fundamentally is that that information is not independent. It has to be not only coming into the brain, but now converging on common sites in the brain where it's integrated and able to give rise to these changes in behavior. Okay? I think Randolph talked about this. My lab considers themselves to be amateur filmmakers, right? Yeah, we all have hobbies. If we talk about the biological imperatives of this, in addition to these things like facilitation of your reaction times, one of the best examples where multisensory systems are most active and engaged is in the context of speech processing. All right? I think Randolph mentioned to you this idea of a cocktail party effect. This is a 40-second, 50-second video. Again, excuse the amateur nature of it, but I think it makes the point very, very clearly in terms of how much of a benefit you can get from having concordant information from two senses, particularly as your world becomes noisier and more complex. So bear with me. So this is first auditory and quiet.
I have 3 classes today. The last one is at 5. Then I have to go to a meeting.
Says Mara Rivera, an undergraduate in my lab. And now she's going to have audiovisual information. You're going to see her lips.
I have 3 classes today. The last one is at 5. Then I have to go to a meeting.
So both are pretty comprehensible. Right? Now we're going to start to really put it in a more complex, noisy environment, the cafeteria. That would be pretty good to pick out what she's saying, right? And
I have 3 classes today. My last one is at 5.
so still difficult, but I think you can see the gain, or you can hear the gain, or you can perceive the gain more accurately, right? And this is really important, right? What people have shown, yeah, here they go with their credits. What folks have shown is that having the ability to see the lips of a speaker in a cocktail party in a noisy environment basically confers effectively about what we would call a 15 decibel gain in the comprehensibility of the auditory signal. Okay, just being able to see those lip movements. So really, really important. Okay. So again, I've alluded to this already. These behavioral and perceptual benefits that we can talk about, they really, to me, they provide fundamentally important insights into what's going on inside the brain that give rise to these changes that we have in our perception and behavior. And again, they tell us that information from the different senses has to converge somewhere within the nervous system. And if we simply look and start to do a laundry list, and this is by no means comprehensive, what we find is that information from the different senses is coming together all the way up and down the brain and spinal cord. Okay, so this is just sort of organized in a hierarchical way. Those neocortical, the new big cortex areas up on the top. But as you make your way down the list, you're moving into areas within the thalamus, within the midbrain, within the brainstem, and then finally within the spinal cord. Okay, so the point of this is very simple, is that information from the different senses converges everywhere along the what we call the neuraxis, the brain and spinal cord. It's a ubiquitous phenomenon. This is just sort of blowing it up. The slide's not terribly visible, but this is cortex, okay, the neocortical structures. On the left is shown a monkey brain. On the right is a human brain. The coloring in that case is showing you, with functional imaging data, what the different convergence patterns are. So the things that are shown in red are where auditory, visual, and touch information come together. The things that are shown in blue, where auditory and visual information come together. The things in green, where visual and tactile, et cetera, et cetera, et cetera. So I think what you can see is, two things I want to point out on this slide is, one, we know that the back part of the brain, occipital cortex, is visual. Okay? We know that the lateral part, the temporal lobe, is auditory. And we know that the parietal cortex, which sort of sits above it, is somatosensory or tactile. Okay? If you notice where these areas sit, they're interposed between those major cortical domains. Okay? So if I want to find, this is the great thing for me as someone who's interested in this. If I want to find an area of the brain that's visual and auditory, what I do is I look at where occipital cortex is and where temporal cortex is, and then I go right between. All right? And what I find is it's chock full of these neurons that do these interesting things that I'm going to talk about next. Okay? So my lab uses a whole host of methods to sort of try to decode and understand a bit better how the brain assembles information from different senses, how it integrates that information, and then transforms it into perceptual and behavior output. Okay, up on the top left is just, in animal models, what we do is we can use very, very fine wires to actually record the activity of individual neurons. Okay, we can listen into the conversations that these neurons are having as we present sensory stimuli. Okay, on the right, EEG is a standard method that we use in the lab, where we can put these series of, in this case now we have 128 electrodes that we can put on the scalp and measure the voltage changes and start to infer the things that are happening inside the brain. And then the bottom are some of the magnetic resonance imaging approaches. Here, what we call functional MR, okay, where I'm looking at activation patterns associated with a specific task. And on the left, a technique called tractography, okay, where what we want to do is we want to really map out what are the connectional patterns of the brain, in my case specifically as they relate to the integration across the different sensory systems. Okay, many of you have probably heard about the Obama Brain Initiative. Okay, it's all about connectomics, trying to really build a connectional map, a road map of the wiring diagram of the brain. Okay, and it's very relevant for what we do. So again, and this is just using these approaches, these are the types of things that we can do, is we can start to put these road maps together and actually then put them into theoretical models or practical models where we can start to say, what are the areas of the brain that are orchestrating the communication between the different sensory systems? All right, that's what this slide on the right shows. It's something called graph theory that we use a lot to sort of weight. It's great to show the roadmap, right? This dense web of interconnectivity. But the powerful thing about connectomics is that not every one of those wires has equal weight. Okay, it's like thinking about highways, right? Superhighways, eight lanes. Okay, rural roads, barely one lane. Okay, that's basically what these kinds of weighting patterns allow us to see. Okay, so let's talk a little bit about, and I'm just going to take you through one slide that sort of illustrates to me, and this is still, to this day when I go into the lab and I see this, it blows me away. What these multisensory neurons do. So again, I showed you that we can record from individual neurons and listen into their conversations, and we can now present a series of stimuli to the animal and now look at how do the neurons respond when we present it with a visual stimulus, an auditory stimulus, and what about when we pair the visual and auditory stimulus? So on this panel on the left, just a couple things to orient you because you're going to see a few of these. All we're doing is I'm taking a very simple bar of light, okay, and I'm just moving it from one location to another, okay, 10 or 20 times, all right? And what we're doing is those little dots up there, each one of those dots represents an action potential. the digital currency of the brain, of the nervous system. This is how neurons communicate. Okay? Each of those dots is aligned in a series of, I don't know if it's a row or a column. I'm horizontally and vertically challenged. So, but basically each trial, you'll see, first trial you may get one spike, the next one you'll get, it's a row, thank you. Yes? This is one, what I'm talking about here is one neuron, yeah. And actually now we're getting more and more sophisticated in looking at networks of neurons, but for this example, we're looking at one neuron. Okay? So we sweep our bar of light, and now we look at the pattern of action potentials, and we can just build a bar graph. And the bottom thing there is just showing you on one trial how this cell responds to that visual stimulus. And it does this, pop, one spike. All right? So it's responding to the visual stimulus. It's pretty puny. All right? This is a multisensor. Yeah. This isn't a rodent, so it's a rat. Okay? In the second example, we're doing the same thing. It's the same cell. Now we're listening into the same cell. And now what we're doing is we're presenting an auditory stimulus. In this case, what we call a broadband noise burst. That's just a tss. That little square wave up at the top shows that. And what you see is that the cell also has an auditory response. Kind of puny, but it has an auditory response. All right, well, here's where the magic happens, right? This is what brings me into the lab every day. Put the two together. Bam. All right? I could make a career like Emerald, right? If I just do this and go, bam! In essence, it goes from a single spike to almost a machine gun burst. All right? What we hear now is when you pair the stimuli, you get cell responds in no way beyond. So put these two together, right? You can add these together and say, what would you predict if the neuron was simply an adding machine? Doesn't in any way come close to what the neuron actually does. Okay, now I want you to kind of remember back to the beginning of the lecture. I talked about these examples in behavior where what we see are things that beat that dotted line prediction. Well, the dotted line prediction is that summing of those visual and auditory inputs, right? These neurons do something transformational. Okay, they take that convergent information and now they build something completely novel out of it. And we believe that these benefits that we get in perception, again, the example where now the cat responds to the bird when we have both stimuli active, this is what's underscoring that. This is what's driving home that behavioral benefit. Powerful for evolution and for behavior. Collectively, we call this phenomenon multisensor integration. Okay? It's a fancy currency name that we give to this, but it's just to reflect the fact that there is this really remarkable transformation that happens when we take information from 2 or even 3 senses and put it together. I don't have a slide for this. I have a slide for another thing, but I want the point I want to make here is that these multisensory interactions, they don't happen in a willy-nilly way. Right? What does that mean? What it means is that they're really very much, they're very smart. Okay? These neurons are very smart. Well, neurons aren't smart, but collectively, we end up being smart because we have lots of neurons. But what they do is they're not just doing these kinds of dramatic enhancements to any stimulus combination we present. Okay? They're doing it only when those stimuli are presented in very specific relationships to one another. Okay? I'm going to use an example. Hey, my glasses, right, they're Walmart glasses, so they're $5 glasses. I'll drop them. I drop them there. 2 things to point out. One is, okay, if I'm looking at it, the visual information associated with that collision with the table, substrate, right, and the auditory information associated with my glasses hitting the table are coming from the same location in space. Okay? So the brain uses that, number 1. Spatial concordance becomes a powerful cue to say, they came from the same location in space, probably belong together. What's the other part of this? Time. All right? When I do that, comes from about the same moment in time. So the nervous system, the brain, is using the temporal relationship, the temporal correspondence, to make an evaluation as to, those things happen close together in time. There's a high likelihood that they belong together. Okay, so those interactions that I showed you at the neural level is that big enhancement that I showed you is when the stimuli were at the same location in space, presented it at around the same time. As I start to separate things in space and time, what happens is that neural interaction starts to disappear. Okay? Another example, another physical characteristic of the stimuli is what we call inverse effectiveness. Right? And this is it's a more complicated slide than it probably needs to be to illustrate this. Okay? I got people's attention. When you have a very effective stimulus in one modality like that, you don't need information from vision to let you know that something happened and even to let you know where it happened. Okay? Where multisensory integration works most effectively is when things are like this. Okay? When each of the sensory systems is providing weak and or ambiguous information. That's when you get this big gain. Okay? Because as soon as you get one that's sufficiently salient, intense, whatever you want to call it, again, multisensory systems, they're not necessary in order for you to know that something happened. That's what this example is just showing. In this case, it's just a woman and her cat turning a head to an approaching dog. Okay, so I've made the point now, and I'm going to use some sophisticated language because this is what we talk about, but it's a very simple concept. The brain is, in essence, a statistical machine. All right? And these examples that I've just talked to you about, that's in fact what we're arguing, is that what the brain does is it uses the statistics of our world, and by statistics I mean something as simple as what I just talked about. Okay, the spatial and temporal relationships, the relative effectiveness of these stimuli, to really make powerful judgments about our world. All right? So in essence, it evaluates that statistical information on the basis of sort of the developmental framework that we have and its experience. Now, I want to, this is just, some of you may have seen this. So how many know Bobby McFerrin? Yeah, so I had the pleasure now of talking at several different conferences with Bobby McFerrin, and there's a wonderful video I'm going to show you talking about what we go on the circuit and argue about. Is Bobby's argument is he's going to show you a great example of what's called the pentatonic scale, right? Something anybody familiar with music is aware of. I'm going to argue that this is a much simpler thing than the pentatonic scale, that this is the probabilistic machine that is the brain. So 3 or 4 minute video, but I think you'll enjoy it. So let's step into it. This was at the World Science Festival in 2009.
Talking about expectations. Watch.
So there's 800 people in the audience.
Okay.
He's fantastic. So I think it's, and go on, it goes on, he talks, they start to talk a bit more about this idea of expectation, right? And again, the pentatonic scale. But for me, and the thing we talk about a lot is, this is a much simpler problem in many ways than relating this to scales. It's what the individuals in the audience are doing is they're making a cognitive map. Okay, and the cognitive map is simply saying, okay, if Bobby stands on a certain part in the stage, right, he started off with sort of an instructive framework. If he's standing here, okay, there's a certain note and tone that you make. Okay, if he moves, the scale ascends or descends. Right? So to me, as a multisensory scientist, right, it's really about the mapping between, in this case, the visual representation of the world, where Bobby's standing on the stage, and the auditory representation. And we do this seamlessly, okay? And again, it's built into this idea that the brain's this amazing predictive machine. It makes these maps fluidly, and it changes these maps. We're learning on the fly these types of relationships, and we do it extraordinarily well. And the generalization, right? He only showed a couple locations, okay? Yet the audience was able to generalize up and down the scale. Pretty wild. The brain is an amazing machine. So, and I'm going to take this a step further, and I'm going to argue, partly because this is my way of rationalizing my existence, is that sensory and multisensory representations are really, they are the building blocks for perception and cognition. Okay? Without them, without accurate information from them, we're going to build corrupted representations of the world around us. And this is going to sort of transition into the little bit I tell you about autism. Okay? So keep that thought with you. So I want to return to one of the concepts just for a few minutes. And it's this concept of time. My lab has gotten really interested in, I've always been interested in time. And we've gotten really interested in sort of multisensory time lately. And I did a sleight of hand with you earlier when I dropped my glasses on the table, right? I said that information was coming to you from the same, at the same time. Or actually, I don't think I lied. I said about the same time. Okay? And it was about the same time. Because vision and audition travel at different rates. Visual information and auditory information travel at different rates. Audition is slow. Right? Vision basically travels at the speed of light. But audition is slow. So rule of thumb, because I don't remember numbers like 330 meters per second. Obviously I did. I'm surprised. But for me it translates into something very simple, which is it's about a foot a millisecond. Okay? So a millisecond is, it's 1,000th of a second. So sound travels 1,000 feet in a second. Okay? Well, if we start to think about this idea of the temporal relationship, right? Well, now we've got a bit of a problem. Because if vision and audition travel at different rates, how does that information then use to statistically bind those things together? Okay? And the answer is, again, the brain is amazing. Right? It doesn't do it on the basis of an absolute moment in time. It creates a window of time in which it evaluates whether those things belong together or not. And we spend a lot of time in the lab sort of, and this is the construct, right? We talk about something called the temporal window of integration. Okay, if a visual and auditory event happen within that window of time, the brain is smart and says, they're close enough together, I'm going to bind them together. As soon as you step outside of that window of time, what happens now is that you get a visual event and an auditory event as separate events. We can study this in the lab by using some very simple tasks. So I can bring you in the lab and have you do a task that we call the simultaneity judgment task. Very, very straightforward. Okay, I'm going to present you a sound and I'm going to present you a light. They're going to occur close together in time. Sometimes they're going to be absolutely simultaneous. Sometimes they're going to be offset from one another. Sometimes vision's going to lead. Sometimes audition's going to lead. And your job is very simple. You're going to push one of two buttons. Were they simultaneous or were they not? And on the basis of that, we can build a distribution that looks like this. This is just simply looking at, so this SOA just means this is the delay between the sound and light. 0 means they're exactly presented at this exact moment in time. This is vision leads, this is audition leads. And then we're just looking at the probability that an individual subject says that they were simultaneous. Well, notice, not surprisingly, that it's peaked at about 0, right? You're pretty good when saying things are simultaneous. But notice, even when things are separated by 100 milliseconds, you still have a very high probability of saying that they were simultaneous. This is this window, this temporal binding window. Yes?
Is there a possibility that these things are going through an area of the brain that's the same, so they're actually in the brain at the
Yes, absolutely. And that's one of the things. Neurons, if I go into that area of the brain that you talk about, neurons have a distribution that looks just like that in terms of their firing patterns. Okay, so they're evaluating, just like you're evaluating the sensory evidence in terms of its temporal relationships, the neurons are doing exactly that as well. Okay, so good, really good question. So we can build these distributions, okay, and then we can use these and we can look inside the brain. Don't even worry about the figure. It's just that we do a lot of brain imaging. And I just want to point out one of the powerful things that we can do with a task like this, all right? So we have this window, and in that individual, that window is about, we called it about 300 milliseconds, right? We're just going to say at about 70%, we're just going to draw a line across there and call that the window. But we have a really powerful opportunity by doing that kind of analysis. Because remember, what happens at, let's say, that 0.7 point, all right, is that means 70% of the time they're calling it simultaneous and 30% of the time they're calling it asynchronous. Okay? Identical stimuli. Well, what we can do is we can look inside the brain and say on those 70% of trials when they called it simultaneous, what does the brain look like versus the 30% when they call it asynchronous? And this is what happens. These areas light up that are doing the evaluation of the temporal interval between the stimuli. Okay, so final part. I have a few minutes left. Everybody's asking themselves, what does this have to do with autism? Right? And we believe a lot. So we actually believe it has a lot to do with a number of clinical conditions. The lab works right now, has projects that are involved in schizophrenia, in dyslexia, in individuals who wear cochlear implants because they have some interesting sensory challenges associated with them. And most importantly, now we're working very, very hard on children with autism. And let me give you the framework for this, is that many of you know autism, right? It's a prevalent neurodevelopmental disability. Current estimates are that 1 child in 68 has it. All right, and that's, you know, there's a big, we could have a lecture to talk about, is the incidence of this rising? Is it really diagnostic accuracy? Is it overdiagnosis? Is it something to do with the sensory world? Okay, that's what I want to come back to. But I think, you know, the DSM-5, which is sort of the Bible for diagnosing autism, its old version, it just came out with a new version that changes these a bit, but doesn't matter for our purposes. It really talks about these children having problems in 3 principal domains: language and communication, social interactions, and then attention, which actually gets into something called restricted interest and repetitive behaviors. Okay, you'll see a lot of the kids, well, they'll hand flap, they'll head bob. If we think about, particularly if we think about language and communication and social interactions, I'm going to make the argument, okay, that these are completely dependent upon sensory information, and they're actually, more importantly from my perspective, they're very much dependent upon the accurate integration of information across the senses. Think about building a healthy reading or language representation. What do kids do? Okay, they learn how to map the graphemic elements of words onto the phonemic elements, the building blocks, the auditory building blocks. Okay? So my argument is that these sensory representations form the foundation, and we've spent a lot of time, neuroscience has spent a lot of time looking at brain areas that deal with social interactions, that deal with language and communication, but maybe we're looking in the wrong place, okay? Because these are being fed by areas that process sensory information. So my argument is that if the sensory information pathways are corrupted in some way, then it's not a surprise to me that these children have problems in language and communication and social interactions. Okay? It's just a natural consequence of it. Let me show you some evidence. So this is the question: could these alterations in sensory processing play an underappreciated and even an important role in autism? So I talked to you about, and I'm going to come back to this, I showed you a distribution. I showed you that distribution for a reason. Okay, and the distribution was just to talk about this temporal binding window. And we can do these kinds of tests. The nice thing about these tests is they're sufficiently simple that we can get children with developmental disabilities like autism to come into the lab, and they actually do these tasks quite well. And now what we can do is we can compare their performance with a completely matched group of children. Actually, we can go so far as we can match them in terms of IQ. Some of these children are very high performing. And look at, are they binding together information from the different senses in the same way as a child who is typically developed, right? I hate that term. I hope I'm not typically developed. Right? But you know, we as scientists, we have to, you can call them controls. Control's a terrible term, right? It just sounds like some test tube in a dish. But they're children without autism is probably a more accurate way to say it. But when we did this, we were shocked. Okay, when we started to do these analyses, what we found is that, in fact, children with autism are binding together information from the different senses differently than our children who are typically developed. And the fundamental difference is that they have bigger temporal binding windows. They're binding together pieces of information over longer periods of time. Now, on first blush, you would say, sounds good, right? Capturing more information and integrating it. But then you think about, as our world is progressing past us rapidly, right? Suddenly now, you don't know which piece of visual information belongs with which piece of auditory information. You're making these inaccurate bindings. It creates confusion. And often what happens, one of the classic features of autism with children with autism is they focus on one sense at a time. And we believe part of the reason they focus on one sense at a time is because that other information is so confusing because it's out of sync. It's out of temporal synchrony with the information from the sense that they focus upon. So the other thing that we found that was really interesting is that as we started to drill down on this, we use a whole battery of different stimuli. Okay, we can start with those flashes and beeps that I showed you earlier. We can do things like the McGurk effect and even more complicated speech-related stimuli. And then we can even do things in between, like tools, hammer hitting a nail, and ask the children to evaluate the temporal relationship between them. Okay? Well, notice what we found is that for the simple flash beep stimuli and for the tools, there were no differences between our groups. Where it emerged was in speech. So these children appear to be binding speech-related visual and auditory information over a longer epoch of time. And we believe, and actually have now shown, that that longer binding window directly relates to scores on language tests, on speech comprehension tests. Okay, so we think it's this building block question. Yes? Yeah, I mean, Asperger's, which we don't use anymore, it's now with this DSM-5, it's now been thrown out. Again, the real differentiator with Asperger's is typically these kids have high IQ. Okay, so they're 90 plus, whereas when we think of autism spectrum more broadly defined, many of those children have what are called intellectual disabilities. Okay, they have IQs that are 70 or lower. Okay, so Asperger's usually has to do with their cognitive performance. Okay. And now, yes, go ahead. Yeah, I mean, I won't talk about it, but that's the next thing that we're looking at is, right, the social piece. Okay, so everything we do in terms of the social cues, I'm going to argue also is an extraordinarily multisensory event, right? So you're not just hearing the intonation of my voice as I go up and down. I can speak like a Valley girl. But you're also watching me wander around the stage, right, wave my hands. All of these things are powerful social cues that children with autism typically have enormous difficulty in picking up on. They also generally don't look at faces, and more specifically, they don't look at eyes. Okay, so that also is a really important piece in the social puzzle, and we believe that some of the multisensory things we're looking at have to do with the reasons that they don't look at eyes. Yes. Nah, actually, great question. I don't know the answer. It's a really good question. I don't know. I don't know if the size of the cohort is large enough to evaluate it. But it's I should know the answer to that, or I should have asked that question, and I haven't. It's a great question. I can tell you, I thought your question was going to go, does the incidence of autism differ across cultures? And the answer appears to be yes and no. And I say that because there was a study that came out 2 years ago in Korea that talked about 1 in every 20 Korean children had autism. It turns out that more recent research suggests that the number really is much more between 1 in 60 and 1 in 70. But there doesn't appear to be a strong cultural difference, which is interesting in itself because it kind of gets at some of the ideological questions. Reason for showing this, again, is not to focus on the details, but just to say, just like what we can do under normal context, we also now are using these tasks that we developed to drill down on what are the brain bases of these changes in sensory processing in children with autism. Okay, that area of the brain that I showed you in an earlier slide that seems to be calculating the temporal relationship between visual and auditory stimuli, appears to be differentially affected in children with autism versus those that are typical, and it appears to happen only with speech-related stimuli. Stay tuned for this, because this is an emerging story from the lab. I'm going to end with a frightening thing and then with a hopeful thing. I've learned that you have to make your audience happy to go away. Yes?
When you said the word hopeful, 8 months ago I woke up and everything I put in my mouth tastes like putrid, rancid. It's a taste thing. I literally can't eat. It makes me want to throw up. And it's been amazing how psychological the sense of taste is.
Yeah.
I can't, well, I do cook for my family, but you want to sit down and have a meal together. It affects, of course, olfactory is the number one indicator for memory.
That's right. Yeah, memory and smell are just remarkable, right? The girlfriend's, the perfume of the girlfriend from 40 years ago that just sets off a cascade of emotions. It's a great, great question. And I think olfaction and taste, because they connect up with the older areas of the brain, often have... You're not going to let me get away with that? So olfaction, right, is coming into areas like the hippocampus and hypothalamus, right, these sort of rudimentary areas. And we believe that they have a stronger emotional valence because they have a connection to these really very, very fundamental structures. Okay? But we can come back to that. Yes? No, dyslexia, we also believe this window that we see is enlarged in children with dyslexia. Okay? And I can talk more about that in the question and answer period. But let me just end with a couple slides again to talk about what's out there and what we believe might be really something that can be useful in the context of autism. So right now, this is the type of environment that is used to treat children with autism. Okay, so everybody, at one level, as a sensory scientist, I'm delighted by the fact that there's an increasing recognition that sensory processing plays a role in autism. I look at a room like this and it scares me to death. Okay? They're putting a developing brain and a developing child into an environment that is extraordinarily cluttered and noisy in the sensory world. Okay, so these kids, they're spinning wheels, there's loud noise, there's changing noises. They rock them on all kinds of swings and devices. And the idea is, it's a really simple idea, right? It's if you habituate, you adapt children, they get better. I don't like that idea, okay? I think that's really frightening because, again, you're doing this in the context of a child who has a developing brain and where changes in the sensory information can be extraordinarily catastrophic in terms of the trajectory that you put that brain on, all right? So that's a worry, and hopefully we're starting to get into the community to talk about what our worries are with this. But the hopeful part is, all of you have heard the term plasticity, right? And plasticity, it's an overused term, I'll be honest with you. So some of you may actually know of things like Lumosity, these brain training programs. Love the idea, all right? I don't necessarily love the implementation. But in the context of what we're talking about, the important point is the following, is that sensory systems and multisensory systems are extraordinarily malleable. They can change, okay? And what we need to do is we need to figure out ways that we can change sensory processing, specifically in children with autism. If we believe that this is a building block question, what we can do is I can take a child, measure that temporal binding window that I talked about, and now put the child, basically what we have in the lab now is we've developed a series of apps for iPads. Okay? And the children come in and they play a game. And they're playing a game and they're having great fun at the game, but that's not really the reason that they're playing the game. They're playing the game because what we're doing is we're training their sensory systems to evaluate the timing between visual and auditory events. And when we bring a child in and we do this, all you have to know is that this broad distribution gets squeezed. Okay? We think that this is really powerful because we've now taken a subset of these kids, this is all work in progress, and we've now looked at their performance on a series of language and communication measures, and they get better. Okay? So again, we really think that the sensory building block piece is a fundamental piece, and if we can train sensory systems in a directed way, right? This is not this blast you in a sensory integration room, but rather say, okay, we know exactly what the nature of the challenge is in this child, and we're going to tailor a program that can actually train their sensory systems to be better at temporally integrating information across the different senses, and hope that this translates and cascades up because of this building block argument. Okay? I just want to thank a whole host of people and a lot of the funding agencies that have gone behind this work, and thank you for your attention. I really appreciate it, and the good questions. Thank you very much. Yes?
It's my understanding that you cannot identify autism until they're maybe 18 months old.
Yeah, 18 months is about the earliest.
So in this training, is there anything that parents can do in the first 18 months? You know, what environments should they be in to maybe start that training whether they need it or not?
Yeah. Yeah, yeah, so the question is, you know, 18 months is about the earliest that you can diagnose autism now. We're getting better and better at this, but are there things, the question is, are there things that can be done early in terms of sensory systems that could potentially then ameliorate the transition to autism? And the answer is, in my mind, and I'm going to editorialize here, because the scientific answer is we don't know. I'm a big believer that part of the challenges with autism and part of this increasing incidence, it's in the diagnostic category, but I also believe that the sensory environment that we place children in can take a child with a genetic predisposition towards autism and can tip the balance. Okay? If they're overwhelmed with sensory information, if their sensory systems are tasked with too much information at too rapid a rate, I think this really can be an important piece in the puzzle. So one argument, you know, what I tell parents that early is, obviously, I don't know the answer, but I would really try to eliminate the complicated sensory environment. You know, you see these, one of the classic examples, we had Huda Zogby here recently, who runs the Autism Institute at Baylor. She has this great slide of a baby infant in a stroller. Mom and dad are on their cell phones, and the baby has an iPad that's kind of put up on the bars here, right? And everybody's confused and looking around. And so that may be a part of the puzzle. Yes? Yeah. Yeah. Did anybody see me give her $20 when we walked into the room? The comment is that this training approach and sort of the work that we're doing, implications beyond autism in terms of typical development, and in this context, specifically in the realm of learning and education. And actually, this is, you know, it's a great plug for, we have the first program in educational neuroscience in the country here at Vanderbilt. And the whole idea is exactly this, is that are there ways that we can build better learning environments for our children that take advantage of some of this knowledge? Like we know, if you put 40 kids in a classroom, all right, it's one learning style. Some children are visual dominant, some are auditory dominant. We can do better, right? And we haven't changed that environment much, and we haven't used specific information, A, about children's individual dominances, but also things like the neuroimaging work, which can tell us a lot about the networks that are processing information. I think this is the next revolution in education, is starting to really use neuroscience concepts to think about building better classroom environments. Follow up, I'll let... Yes, I mean, 20% of people shift dominance during life. What seems to be interesting about that is the work that shows it is that the typical transition is from vision to audition, and it seems to be related very much to musical experience. Is that as children become more and more proficient in music, what happens is they shift their dominance base. Okay. Yes?
What do you say to still claim that vaccines?
Yeah, there's no evidence that vaccines, I think, you know, and that, it's unfortunate that this has gotten such high profile, but I've looked through all of the literature and evidence, and there's absolutely no link that we know of between vaccines and autism. Yes?
What exactly does the exercises for narrowing the perception temporality consist of? You said that he was doing it, but what...
Yeah, they're games like, yeah. So here's an example: is you're a soccer goalie. Okay, one of the fun ones that we have now is, and what soccer goalies are very good at doing, believe it or not, is they're good at reading the cues associated with not just what they see, but also the contact point of the ball with the foot. And so what we're doing is we're basically having children play as goalies, and then we're using information from vision and audition to have them make assessments about where the ball is going, the trajectory of the ball, and using that to sort of shape the temporal binding window by giving them examples, giving them good feedback in terms of, okay, you got that right because you integrated this over this period of time, or you got it wrong because you integrated over too broad a period of time, right? One example. Yes?
When reading orally, some people use vocal inflection. Others don't seem to be able to do that. Is that all combined with this multisensory?
Yeah, I think that's another interesting, audition itself has, all of the modalities have these sort of submodalities. This is one in terms of what we call voicing, right? There's certain degrees of voicing that have a high degree of individuality. And these things are also a part of the integration puzzle is, how do we, one of the great speech mysteries is everybody in the room has a different vocal tract, okay, and articulates words differently, but nonetheless, we will have no trouble understanding that. So this is the question of integration, is how do we take that different voicing information and yet build it around, this specific phoneme or this specific word. So that's what I would call within, I've talked about multisensor integration today, but there's also a lot of integration that goes on within a sense. Right? Best example is vision. Right? If you think about vision, what it's doing is it's integrating form, it's integrating motion, it's integrating color. All of those are sort of submodalities that have to then be put together in order to say that this is my cell phone. Yes? Yeah. I mean, in terms of some of the auditory, so there's a lot of work that's done on what's called phonemic contrasts, which is basically these differentiation between one phoneme and another. Well, it turns out generally Asian cultures, Chinese is the one that's been studied the most. They are much, much better at some of these phonemic contrasts. Again, because the same basic character, depending on inflection, can be 5 different words. Or 5 different meanings. So they're more acute at picking up on those subtle. We as Westerners don't pick up on those cues at all. Yes. Yeah, we learn again that this binding window, particularly in this case, I'm sorry. The question is about dyslexia. In this case, is what have we learned about dyslexia, like what we've learned about autism? The mystery for us is that, much like with autism, with dyslexia, we find a larger temporal binding window. Obviously, children with dyslexia don't have autism, right? So that's the sort of disconnect for us now, is that it's clear that that temporal binding window is only a piece in terms of how you build the higher order representations. What we think is that, for some reason that we don't yet understand, is that integration, that temporal integration, is more important for reading representations in those with dyslexia. Yes? Mm-hmm. Yeah, so the musical training, it turns out that there's 2 groups that we bring into the lab that we always are blown away by because they perform off scale on a lot of our tasks. They are musicians and athletes, okay? Both of whom seem to have, and in somewhat different spectrums, but they have really, really tight temporal acuity. These binding windows that I talk about, they tend to be narrow in these individuals, right? And you start to think, okay, what are the consequences of having too narrow a binding window? We have folks in the lab where there's in particular one Vanderbilt baseball player whose binding window it's about 70 milliseconds, which is 3 orders of magnitude off scale compared to, or 3 standard deviations off of what we do. We think it's it's a genetic underpinning, and then the experiential relationship of a lot, 6 hours a day of baseball practice make you really good about evaluating the temporal relationships between what you hear and what you see. It goes back to the soccer player question. Music seems to do something very similar, okay? That there seems to be an ability for them to narrow the temporal binding window.
What happens when the person stops being a musician
Yeah, that's a good question. I don't know if anybody's followed up on it. That would be really interesting to see what they do. I don't know if this is something that, once you set it in stone. My guess is we know that there's adult plasticity, both good plasticity and bad plasticity, but it's slow. So my guess is that they'll stay pretty narrow for a while, but then over time it'll start to creep back out. But I don't know. It's a good question. Yeah. Really? Fantastic. Mm-hmm. ADHD, yep, that's right. That's fantastic. That's a wonderful story. Thank you for sharing. Question?
The exclusivity of Vanderbilt being in the position of having this information, how do you share that now with other organizations in this country?
Yeah, so we, talking about educational neuroscience in particular, so we are now going on the road, a group of us, to national conferences to talk about what we have here in this space and how we're trying to really knit the pieces of neuroscience and education together. So we'll go to, one of the wonderful things here, come back, editorialize for a second, is Peabody is the best education school in the country. Okay? Our neuroscience program is the best neuroscience program in the country. We have the natural synergy. And now what we want to do is we want to go to others and challenge them. We want to go to educators and challenge them to think about it in this context. We want to go to neuroscientists and say, we need more of this, right? This is about neuroscience and society. In my perspective, one of the things I've gotten very interested in the last couple of years is creating programs at the intersection between neuroscience and society, because we now have technology that really allows us to make decisions about things like educational style and learning strategies that we didn't have 10 years ago. And we need to use that in order to really better the society that we live in. And I think that's my bully pulpit these days, is that this isn't just for us to toil in our labs. We really need to be able to tell the world how this work is so relevant for what they do. Yes? Yes. Yep. Yeah, Kennedy Center is one of the major partners in this. Absolutely. Yep. Yeah. Yes. Yes, actually a very strong link. It's a great observation. So I have a slide. I didn't show it. It's a slide of historical synesthetes, right? And if you look at people from Galileo, right, there's a lot of evidence that Galileo was synesthetic. The problem is it sort of falls apart when you get to Tom Cruise, right? So he shows up on that slide as well. But no, there seems to be, it's a really good observation. And it comes back to a point that I raised, but I didn't make strongly enough, which is this connectivity issue, right? So I told you that synesthesia appears to be the fact that there's these connections that aren't pruned. Well, one of the things that seems to be emerging from studies of synesthetes is that that connection isn't local, it's actually more global. So they have more neural connectivity over the entire brain than do other individuals. And this may be the basis for this idea of creativity, enhanced creativity in these individuals. One interesting thing is that David Eagleman, who I mentioned earlier, who does the synesthetic battery, he has an interesting series of imaging studies now that are saying synesthesia and autism are two ends of a spectrum of human brain connections. That these are children with autism, and we know this, it's called an underconnectivity disorder. Synesthetes have overconnectivity, and that what we're looking at is we're just looking at those two global ends of the distribution and how they manifest. Interesting idea. There's still some weaknesses in the argument, but I think it's a kind of cool way to frame how we think about brain connections. Yes?
I've heard that the children learn language easiest because we do baby talk. And is that the same thing?
Absolutely. Absolutely. Any of the motherese, it's all about enhancing a lot of the visual cues, changing that level of intonation, much more exaggerated way. All of those things are really so that the babies are sponges, okay? And in fact, one thing I didn't tell you is this temporal binding window. Early in development, it's like this. Okay? Babies are sucking in all the information. And then what happens with time is this. right? And that's, Mother Ease is a great example of that, is that early on with all of those exaggerated prosody information is that the babies are just, they're making those statistical relationships and then they're narrowing them down over time. So I'm glad you raised that. It's a really good point. Yes? Yes, yeah. I mean, I think all of these things ultimately boil down to changes in brain connections. In schizophrenia, in this case, I think what it is the level of the connectivity change is different in terms of there are areas of the brain that are just simply wired either less or differently than there are in, let's say, a child with autism that appears to be a much more global connectivity disorder. So I think everybody who works in the field of psychiatry will tell you that whether we're talking about schizophrenia, we're talking about bipolar disease, even depression, that there's a fundamental aspect of changes in connectivity that underlie that. But then you get into this sort of, is it a generalized effect or is it an effect that's talking about communication between specific brain regions? And that's where I think you get into schizophrenia being a bit different from autism. Yeah, no, I mean, that's the other thing is the chemicals. So the question is, does it have anything to do with the chemicals? And obviously we know from work on things like selective serotonin reuptake inhibitors, SSRIs, Prozac, right? That serotonin is a major neurotransmitter, appears to play a powerful role in things like depression. But the missing piece in the puzzle is that these neuromodulators, neurotransmitters, also shape connectivity in powerful ways. So that's the sort of the taking it from the molecular level up to the systems level is that serotonin will change brain connectivity. And that may be more important than the fact that it's the serotonin itself. Thank you all. Thank you.